Okay, so I'm going to cover two things. Firstly, the title of the talk is the Enteric Virome, and I want to introduce what that actually is. We tend to think that the viruses that we have within us, we only have them when we're ill and we're infected with a virus that causes disease. It's actually the case that we contain many viruses and very large numbers of them in the gut. And it's only recently that the technology has allowed us to look at these in detail. And then I want to, at the end, describe a sort of an ongoing study of patients with chronic fatigue syndrome, and we're looking at the virome in their intestines. So, the gut contains an enteric virome. So what is this enteric virome? Why is it important? And how can it affect disease? And the concept I want to give over here is that a large amount of data accumulating on genes involved in disease, environment and disease, all sorts of things affecting disease, and that particularly for diseases which seem to be caused, multifactorial diseases, there's probably multiple causes. And the sort of omics technologies that we have these days, the big data sets, allow us to look at people in great detail, but then you need some sort of a hypothetical framework as to how all that data can be used to understand disease in more detail. Okay? So this is the gut. As you all know, it's a very large organ within your body, and it is full of bacteria. Okay? So the gut is actually a very important part of our immune system. So it contains billions of bacteria called the microbiota, and it has its own immune system, but it ignores our own bacteria. So we are full of bacteria, and yet we don't make an immune response against it. Okay? And that's because there's a sort of a homeostasis between all the microbes that live in the gut and then this immune system that surrounds the gut and somehow can tell when a bad bacteria comes through. It will give you an immune response against it, but at the same time, it ignores these trillions of bacteria that live in our gut. Okay? So the recent work suggests, and Joe's slide as well had this, immune system, gut-brain axis. So really, the interaction between the host immune system and the microbiota can set an inflammatory threshold which can influence disease. So we have the bugs that live in the gut and the immune system, and that gives you, each individual, an immunophenotype. So this interaction is generating cytokines, other signaling molecules from the immune system, which circulate through your body, and then they can affect other organs. So they basically set a thing called an inflammatory threshold. So we're not inflammatory null. We have what is a useful threshold which can respond quickly to infection, but under normal circumstances doesn't attack your own tissues. If the inflammatory threshold is upset, you can be predisposed to many diseases: depression, inflammatory bowel disease, diabetes, obesity, metabolic disease, cardiovascular disease. And many of these studies have been underpinned by mice models of disease, which don't get the disease unless you put those bugs back into their intestines, all right? And when you put the bugs into the intestine, the inflammatory threshold is raised, and then you get disease. So this is the sort of idea we are approaching by looking at chronic fatigue, ME. Okay? So you can be predisposed to this depending on your host genes. Okay? So you can have a genetic predisposition to this, and this would be a reason for looking at all the host genes and mutations in patients with chronic fatigue. You might be able to relate a genetic predisposition to the immune response, and the inflammatory threshold. So one factor is your own genes. Okay? The other factor is genes contained by viruses which also infect the gut. So most of the work up until now has looked at bacteria in the gut, but recent methods allow us to look at viruses. And it turns out that the gut contains large amounts of viruses that are viruses that infect our tissue, but they also contain a larger number of viruses that infect the bacteria that live in your gut. Okay? So each microbe in your gut lives in balance with a virus that will try and infect it. So these are also affecting the interaction between the bacteria and the immune system. And we've only recently found methods to actually study these viruses. Okay? So here they are. We've got the enteric viruses. They can affect the microbes in the gut and therefore affect the interaction between the microbes and the immune system. And they can also affect the immune system directly. So they're an important part of setting our inflammatory threshold. Okay, so human viruses can affect the immune responses, and examples of these are RNA viruses such as poliovirus, Coxsackie virus, retroviruses, norovirus, reoviruses. When people sequence the viruses in the gut, they find remnants of all these types of viruses there, even though there's no signs of infection, and there's DNA viruses as well. So we've got viruses which affect humans, can affect the inflammatory threshold, and then there's the viruses which infect the bugs, can also set the inflammatory threshold. Now, biologically, these are very interesting because these things are called phages, and they probably evolved billions of years ago as parasites of bacteria before there were any animals on the planet. And there's two main types of these ones that really do look like spaceships. They land on the bugs with a landing platform, and when they hit the surface, they use this bit here as a syringe to inject their genome into the bacteria. Okay? And these are called the lambda phages. And then there's smaller ones called the microvirale, basically small viruses, and the lambda phage are the large viruses. Okay? So the idea here is that these phage can infect the microbiota, and they can do two things. Firstly, if they kill the microbiota, they might kill the good bacteria, leaving the bad bacteria there. And I hope the next slide, no, I'll do with this later. So what we have here is the virotype hypothesis, and this was proposed last year and the year before by some groups in the States, so that when you're looking at a disease, you've got your own genes, which is your genotype. You've got the genes of all the viruses to consider in combination with how they interact with the immune system and set an inflammatory threshold. So this is being investigated in a large number of ways by whether or not this inflammatory threshold affects diseases, and also it can affect vaccination, and it can affect your susceptibility to microbial infection, and it can be influenced by the environment. Okay? So we got interested in this because part of the work on my lab looks at inflammatory bowel disease, and actually some work has shown that if you affect the balance of bacteria, microbiota in the gut, you upset the gut, and then the bacteria move into the immune system, create an inflammation which upsets the inflammatory threshold. Okay? And work last year showed that in inflammatory bowel disease, that is Crohn's disease and ulcerative colitis, the balance of the viruses in the gut changes. Okay, so they look at all the patients, they find that the diversity of the bacteria in the gut goes down, and the diversity of the viruses in the gut goes up, suggesting that there's a relationship between the disease, that is, increased inflammation in the gut, and the balance of the viruses in the gut and the microbiota. Okay? Now, the interesting thing about some of these viruses is lambdaphages, they inject their genome into the bacteria, and that genome then enters the genome of the microbe. So not only can they kill the bugs, they can affect what the microbes make. And these genes affect the metabolism of the bug, allowing the bug to generate all sorts of metabolites it wouldn't normally make, and those metabolites can also affect the immune system directly. So the work that we did was sponsored by Invest in ME. It involved Simon Carding, who we'll talk later, who's gut biologist and immunologist. Amalak Bansal gave us access to patients. Dan Vipond, a PhD student funded by Invest in ME, has consented patients and got samples. And the samples we take are fecal samples. It's just a small sample of feces. And then Maureen Hanson at Cornell agreed to take a medical student sponsored by Invest in ME to her lab to show Navina how to isolate viruses from fecal samples. And then I'll explain how the process works. And then you basically isolate viruses, sequence them, and then you need bioinformaticians to work out what's going on. So how do you find all the viruses in your gut? Sounds like it's a big project, but actually it's fairly simple. You take a fecal sample, just 100 milligrams, and you suspend it in water, and you've got all sorts of stuff in there. You've got dead cells, you've got bacteria, you've got viruses, and you've got food, and you've got goodness knows what. But most of that stuff is very large, and viruses are very small. And the first experiments that identified viruses at the end of the 18th century showed that if you had a filter with a very small space in it, you could pass infected material through the filter. All the bacteria stayed on top of the filter, but infectious agents came through, and those were the viruses. Because they're very small, they'll pass through the filter. So that's a quick isolation of the viruses. And so any genetic material that flows through here, if it's contained within a capsid, that is the protein box which makes up the virus, it's protected from enzymes that would degrade the genetic material. So we filter the feces, take the very small particles, digest any DNA or RNA which is outside, and assume anything that is left in a protein box and is pelleted is virus genetic material, and then we use powerful sequencing machines to provide all the sequences. And then you need bioinformatics to sort it all out. So we've thus far looked at 16 fecal samples from 16 moderate patients suffering from moderate chronic fatigue. Each one gives us 2 million readouts, so we get 2 million data sets from each patient. And these have to go through a whole series of bioinformatic quality filters, and that reduced that to 700,000 useful samples. And then you can use software which allows you to piece all the bits of DNA together to give you a virus sequence. So this is just one patient from 100 milligrams of feces. Okay? So here is all the aligned DNA sequences. About 3/4 actually turns out to be the sequences of the bugs that live in the gut. So some of their genetic material is getting through, and we can see all these different bugs here. Some of this is viruses, and then we can look at these sequences, and we can see we can separate out the different virus families. These are those big lambdaphages, and these are the small microbes the smaller phages, and then we can actually separate this out into the different phage families. Okay? This is quite a lot of bioinformatic work, but the really interesting thing is that each flea has a flea that lives on a flea, if you look at it this way. So every different bug, so the microbiota in the gut is 25 or 30 different families of bacteria, and each family has its own phage, all right? So this actually allows us now to go into all the phage sequences and assign the phages to each member of the bacterial families in the gut, okay? And that's where we are right now. So I say this because the work in other diseases is moving in this direction, and people hope to be able to correlate an imbalance in phage populations with disease, and that has an effect because those phage genes go into the bugs that live in your gut and change the way they metabolize. And if you think each one of you has 1 trillion bugs in your intestines, this is an enormous genetic influence on the bacteria that live in your gut. And really, the next stage is to actually see how do these phages influence the metabolism of the microbes that they infect, and how do the phages affect the populations of different bacteria. Now, it's very difficult to think how this would end up as a therapy, but actually, if there was a correlation, it's a very quick biomarker. I mean, you just need a fecal sample. It's all the patient would need to do is provide a fecal sample. We can isolate the viral DNAs, the bacterial DNAs very quickly, and you can get a complete genetic workup on what's going on in the guts of those patients, and that's really our goal right now. And none of this would have been possible without Pia and Richard's interest in our work at UEA, Maureen Hansen, virus isolation, Amalak Bansal, providing patients, classifying them and recruiting them, and Leslie Hoyles doing the bioinformatics. And this is Dan Vipond, sponsored by Invest in ME for a PhD, and 2 medical students who have done master's degrees, again, with sponsorship from Invest in ME. And the idea here is to get new medics interested in this disease. If we can get the medical students on board, then they'll go on being interested in this disease as they go through their careers. I'll leave it there.
Thanks, Tom. Well, I'm sure you've never thought about your gut in those terms before. It's novel to me. I'd forgotten about phages, and it was interesting to show you how complex it really all is, and unravelling the effects is going to take time, support, and numbers of people, but you can see they're on their way. So, questions, please? Yes, there's one up there on the left, Pia. Anybody on the right? I can't see. No. Yes, and one down the front here, yes. Yes, one up there, yeah.
Thank you. I noticed in one of the slides you said there was an overpresence of tomato in the, as well as, I can't remember what the other thing was. My question is, considering a lot of people have intolerances, food reactions, particularly celiac with ME, has your research separated out whether some of the patients that you're testing also have consideration of either intolerances or non-typical celiac or celiac itself?
Oh, yeah, I think that's a good question. So if you're thinking about the other autoimmunities, or the other immunities, it's this inflammatory threshold. So if the threshold is raised, you're more likely to have antibodies made against other things. So it may be possible correlating all this data with different diseases and people with different types or complexions of the same disease, all thinking in terms of this inflammatory threshold setting your immune system, not only that, but inflammation in other organs, including the brain.
There was another one, sorry.
I've got a son who's been ill for 10 years, in his 30s. I've seen Amylec cell. I do believe it's very much in the gut. But how do you, when you find all this, how do you actually treat it? What's the end
I
treatment?
guess that really is, that is the difficult side of this. Now, we had a talk yesterday about how the bioinformatics and everything can be used through computation to think of new therapies when you know everything that's wrong, all right? So it may be, so this could say, yes, the inflammatory threshold could be high, and then there would be thoughts about a drug that in part would reduce an inflammatory threshold, but that won't be the entire story, because you would have to think of other types of therapy to affect the other symptoms. So what I think the point here is that this is a very complicated disease, and it's not easy to think about how any one investigation could cause a cure until you pinpoint a single cause. So,
but if you have your investigations, you get all the results from that. Is there a way then to go forward and treat the gut?
Well, you could consider probiotics maybe. There are ways of engineering bugs to suppress the immune system in the gut. But I think we're a long way off a therapy here, but at least it's a fairly rapid assay for, do these patients have a consistent change in the composition of their microbiota and their viruses, and could that be affecting their immune responses?
So where did we get our bugs from in the first place? Is it our mothers?
I think so, yes. I think they come from
Yeah. It's all your fault again, you see. You'll get the blame anyhow, you know. But seriously, I mean, it's quite an interesting area, I think, of research. But
It's
complicated.
also that this population of microbes changes quite a lot over the first 18 months of life, and then it seems to be set, and you're with it for life. And you carry that genome of microbes with you as your personal extra genome.
It's part of your inheritance, in other words, besides Mendelian genes in your genome, you know. And it's a new way of thinking about it all. It's been around for a while, but it's becoming serious research now, and the ME is part of it, which is big, you know, another move forward. There was one over here, the gentleman over there, yes.
What can fecal transplantation tell us about what you're working on now?
It's actually the interesting issue that came out with the experiment with the inflammatory bowel disease was that you've got to consider these basically the phages which infect the bacteria when you do a fecal transplant. Because if you do a fecal transplant and you kill all the microbes, the bacteria with antibiotics, you're not killing the phage. So when your fecal transplant goes back in, it will be probably infected by your own phage population. So it's definitely a consideration.
But surely
the bugs you put in will bring their own phages, and then you end up with a fight.
Yeah. So phages could potentially be competitive.
Yes, they are. And I mean, there are actually, in the 50s and 60s, Russia did a lot of work on phage therapy because they wanted to use it instead of antibiotics. And that all fell by the wayside, but it's resurging now because much more is understood about how to engineer phages to do what you want.
There's a fantastic documentary program that you can see on YouTube on that very subject about what the Russians did back in the 50s and 60s.
Well, Tom, thank you very, very much indeed.